Hello everyone. In this video, let us get a glimpse of compound microscope. Here, we are going to gather knowledge about the construction and working of a compound microscope. A microscope is an optical instrument which is used for viewing very small objects such as animal cell, plant cell, and so on, which typically magnifies the object several hundred times. Microscope contains multiple lenses which helps in collecting light from the object and then in focusing the light into the eye. Microscope has the power to magnify very minute objects to a considerable size, giving small to smaller details. Now, let us discuss the parts of microscope. A microscope has three structural components. The first, which is the head or the body. It comprises optical parts in the upper part of the microscope. This is the base which supports the microscope, and it has a mirror within it. Next, the arm. It connects the base and the head of the microscope, and it also allows the body of the microscope to tilt. The next which we are going to discuss is eyepiece. This is what we look through at the top of the microscope. A typical standard eyepiece has a magnifying power of 10x, but varying powers are also available. The next is going to be body tube of microscope. A body tube is comprised of two smaller telescopic tubes with lenses which are mounted at the either ends. The wider tube holds the turret with objective lens, as we can see with a second bracket, and the narrower tube which holds the eyepiece. The next topic which we will discuss is objective lenses. Objective lenses are primary optical lenses on a microscope, and a typical one includes 3 to 4 lenses mostly. Next, we will discuss about the coarse and fine adjustment knobs. These knobs are used to focus microscope. The knobs help to slide the body tube one into another by varying the distance between the lenses. The bigger one is the coarse adjustment knob, and the smaller one below that is the fine adjustment knob. The next topic is turret of microscope. It is nothing but a revolving nose piece which is known as turret, and it is attached to the lower end of the objective, and the turret can be rotated to change the power of magnification as required. The next part which we will discuss is stage clips and aperture. Below the objective, there is a flat platform which is known as the stage. There are stage clips which hold the slide with specimen in proper place. A small aperture is also present in the stage which allows the light into the body. The next part which we will discuss is mirror. A small mirror at the base of the microscope reflects the light through the stage aperture. Next is diaphragm and condenser. Diaphragm is present below the stage and controls the amount of light reaching the specimen, and the condenser is located below diaphragm and is used to collect and focus the light from the illuminator, which is generally our sun. There is a knob also beside the condenser which helps it to move up or down to control focusing on light. The next part which we will discuss is lens. The lens is present, one at the eyepiece, one at the objective. These lenses are biconvex with different focal length. The objective lens has a short focal length, whereas the eyepiece lens has more focal length. Now we will discuss about the working principle of a compound microscope. Here the object O is placed just outside FO, which is the focus of the objective lens. A real inverted and magnified image I1 is formed. It acts as object for the lens, eye lens. The eyepiece is adjusted such that the image I1 lies within the focus of the eyepiece FE, focus of the eyepiece lens. The eyepiece functions as magnifying glass and forms a virtual, erect and magnified image I2 of the object. To calculate the magnification, it is the total magnification, which is the multiplication of the magnifying power of objective with the magnifying power of eyepiece. Magnifying power of objective is represented as MO and magnifying power of eyepiece is represented as ME. So let's say objective lens is having a magnification of 40x and an eyepiece magnification lens is having 10x. So the magnifying power is going to be 400x, right? So to summarize the discussion, compound microscope with a system of convex lenses causes magnification of minute objects to a very large image. With this, we come to the end of this discussion. Thank you.